In our next lab, we're going to work with bacteria and give them some pretty special powers through the process of transformation. Let's take a look first of all at what the purpose of this lab is. The main goal is to transform bacteria with a gene from a jellyfish. And we're going to use a special genetically engineered plasmid called pGLO in order to do that. And we will investigate how our efforts at transformation affect the traits of the bacteria. We also want to investigate how the environment affects gene expression. So in other words, we're going to manipulate the environment to turn on and off genes. In this video, we're going to take a look at the basic concepts that you'll need to understand the lab. So all of the notes that you take from this video will comprise the background section of your lab notebook entry. Let's start with a quick review of E. coli bacterium. Remember that E. coli are prokaryotes, meaning they don't have a nucleus. They just have one large circular chromosome. Plus, they've got special pieces of DNA that are also circular but much smaller called plasmids. And plasmids can be transferred from one bacteria to another in order to increase genetic variation. In lab, we're going to be growing bacteria. Another word for growing bacteria is to culture it. And the main way to grow bacteria is to use a petri dish like this and have a jelly-like substance called agar on it. The agar is really just a growth medium. It provides nutrients and a place for the bacteria to grow. And oftentimes, bacteria will grow in colonies. Each colony or clump that you see on a plate contains millions of bacterial cells that are all identical to each other. However, the colony here might be a little bit different than the colony here. Let's talk a little bit more about what goes into that agar that the bacteria are growing on. In this particular lab, the agar contains a nutrient broth called LB broth, and this is full of carbohydrates, amino acids, and all sorts of vitamins and minerals that bacteria need to survive. And it gives the agar a little bit of a yellowish hue. Without this nutrient broth, the bacteria wouldn't have the materials they need to live. Some of the plates that we use are also going to contain ampicillin, which kind of sounds like penicillin, which might lead you to figure out that ampicillin is an antibiotic. Its job is to kill bacteria and other living organisms. So if a plate has ampicillin in it, like this plate, most bacteria will not be able to grow, which is why you don't see any colonies, nothing white on this plate. However, if there's no ampicillin in a plate, like this one, the bacteria will be free to grow because they've got space and nutrients to grow on, but there's nothing there, the ampicillin, to kill them. And in this case, because there's nothing restricting their growth, we get a lawn of bacterial cells. A lawn is when there's a wide expanse of bacteria, just like the lawn you normally hear of is an expanse of grass. Transformation. We've learned about this in previous lessons. Remember that transformation is the uptake of naked DNA, such as plasmids, into a bacterial cell. And this is our goal. We want to make the bacteria that we grow in lab take up some very special plasmids. And let's take a look at the special plasmid we're going to be working with. The pGLO plasmid has some very important features. Remember that the pGLO plasmid is entirely composed of DNA, but that DNA could be used to build special proteins. And this pGLO plasmid does not exist in nature. It was genetically engineered by scientists using restriction enzymes and ligase so that they could cut and paste together different genes specifically for this experiment. So one of those genes is the GFP gene. And this is a gene taken from a jellyfish. And we're going to hopefully put it into the bacteria so that the bacteria can use the gene to make green fluorescent protein. And as you can guess, the green fluorescent protein will make an organism glow green in the dark. Now, we learned that prokaryotes often use operons to control whether their genes get expressed or not. There is an operon that is controlling GFP in this plasmid. and that involves ERA-C. This is a sequence of DNA. It's a regulatory gene, and it codes for the ERA-C repressor protein. And that repressor protein is going to act as a switch, which could either turn on the expression of GFP so that the green fluorescent protein is produced, or it might turn off the expression of GFP. And then even though the gene is there won't be any green fluorescent protein. There's one more very important gene in this plasmid, and that is the BLA gene. BLA codes for the protein beta-lactamase, and this protein provides the bacteria resistance to ampicillin. So if a bacteria can produce beta-lactamase, because it's got the BLA gene, it will not be killed by antibiotics, at least not by ampicillin. So let's take a closer look. Right now we're just going to zoom in on this part of the plasmid, the operon that contains the special GFP gene, so that we know how that works. So here's the GFP gene. This is the structural gene that will code for green fluorescent protein. In front of it's the promoter, and that's where RNA polymerase has to bind. Now, normally, because this is an inducible operon, the ERA-C protein, our repressor, is bound near the promoter, and that prevents RNA polymerase from transcribing and translating. So normally, GFP will not be produced. However, if we put some arabinose in the environment, arabinose is a type of sugar, the arabinose will bind to ERA-C, and you can see that the ERA-C has changed shape a little bit. And this conformational change turns ERA-C from a repressor, which it was in this situation, into an activator. And now ERA-C as an activator will help RNA polymerase bind to the promoter and transcribe and translate the GFP gene. So the bottom line here is that arabinose is necessary for the bacteria to glow in the dark. Without arabinose, even if they've got the GFP gene, they will not express the GFP protein. So let's put all this together and take a look at what that plasmid would do to an E. coli cell. So here we've got our plasmids. This might be the startup lab. Hopefully, we'll get the bacteria to take in one of those pGLO plasmids. Now remember that pGLO itself is just DNA, but the DNA can be used to express this protein, beta-lactamase, which will help the bacteria survive in ampicillin. If arabinose is around, the plasmid will also be used to express green fluorescent protein, which will help it glow. So these are the two powers that we're going to try and give bacteria: the ability to glow and the ability to grow if ampicillin is around. And the reason why ampicillin is being used is because it helps us screen for the E. coli that have actually been transformed. So here is an E. coli cell that did not get a pGLO plasmid. It was not successfully transformed. And as a result, if we grow it on a plate with ampicillin, well, it won't grow at all. It will die. And so we'll be able to tell by looking at this plate that no bacteria were transformed, none of the bacteria got the plasmid. Whereas in this bacterial cell, it did successfully take in the plasmid. It was transformed, which means if we put it on a plate with ampicillin, we will see some growth. So that tells us, looking at this plate, that all of these bacteria must have gotten the pGLO plasmid, because without the pGLO plasmid, it wouldn't have that ampicillin-resistant gene. So we're going to conclude just by looking at an overview of the experimental design. In our next lesson, we'll look at the protocol in more detail. But here's basically what's going to happen. You're going to have a plate full of bacteria. Let's back up a little bit. All right, and these are just regular, normal bacteria. As you can see, they're white, they're not glowing, there's no ampicillin, they're not doing anything special. And you're going to take the bacteria, and you're going to mix some of it with pGLO plasmid. That's going to be your experimental group. You're going to mix other bacteria with a solution that does not contain the plasmid. That's your control group, the normal situation. And then we're going to grow them on different plates. We're going to culture them on these plates. So you'll take your pGLO plasmid plus bacteria, and you're going to put it on a plate that has LB, the nutrients, plus ampicillin, the antibiotic. You're also going to put some of this on a plate that has LB, the nutrient, ampicillin, the antibiotic, and arabinose, the sugar that is needed for the bacteria to grow. And then you'll take your control solution. Remember, this is just E. coli, no plasmid. And you'll put it on a plate that has the nutrients and ampicillin, and you'll put it on a plate that has just nutrients. So you can see from looking at this that LB is one of the constants. All the plates have to have nutrients. Another constant is the E. coli. You're going to be putting E. coli cells on all of the plates. What's different is, do those E. coli have plasmid or not? And how does that affect their ability to grow and glow on these different plates? And during class, we will make a hypothesis about what you expect to happen before we actually conduct the lab. So I will leave you on this note and remind you to make sure that you put all your notes into the background section of your lab entry, and there is a poll to complete.